So good evening, and welcome to this evening's SLAC public lecture. Last month, we celebrated the 10th anniversary of the first beams at our X-ray laser, which is out there in the SLAC Accelerator yard. Those of you who've been here before have heard a number of lectures about results from this machine, but it's a big deal now. It's 10 years. We had a celebration in April, and actually these couple public lectures surrounding that are devoted to results from the LCLS, the X-ray laser that we've created here. Today's speaker is Sébastien Boutet. Sébastien grew up in Quebec, went to McGill, did his PhD work at the University of Illinois, and that work involved the study of the structure of protein molecules using X-ray diffraction imaging. That gave him a little taste for the subject of biological imaging with X-rays. And as you'll see, this device, the X-ray laser, allows us to be kind of a mecca for that study. And so finishing his PhD, he came here. He joined actually what was the lead-up effort to that, which took place at the DAISY laboratory in Hamburg, and at the same time began developing the equipment which is needed to explore the structure of all kinds of different biological molecules here at SLAC. So without further ado, let me introduce Sébastien, and he'll tell you all about that topic. Sébastien, thank you very much.
Thanks for coming. The plan for me today is to try to give you, at the end of this presentation, give you an idea of what's behind this complicated, pretty picture here at the front. There's lots going on here, and my intent here is for you guys to walk away knowing all the little parts that make up an experiment that allows us to study biology in action. So, just as a quick introduction of the main goal of what we're trying to do here in this field is we're trying to see the molecules of life in action. That is what we're trying to do, and we're trying to use the X-ray laser to achieve that goal. So what is shown here is a collection of biological molecules that we're all made of and all life is made of, and some have very important roles, and everything like we all are, is made of atoms, and we're trying to understand where the atoms are, what they are doing, and try to deduce from that what is the function of all these molecules that keep us alive. So examples here that are pretty obviously well known by mostly everybody is a virus. Everybody knows that viruses can be bad for you, but our body reacts by creating antibodies to counteract the actions of viruses. Other molecules shown here are actin and myosin, that are molecules in your muscles that perform actions that contract and expand your muscles that allow you to move. And here, a ribosome is a molecule that makes all these other molecules, makes all other proteins by interpreting DNA and RNA. So this is what we're trying to do, trying to see where the atoms are, what they're doing, and try to ultimately make movies to understand the function and biology in action. So what is shown here is a simple lock and key model of interactions between biological molecules, where you may have a certain molecule, potentially something that does something bad in your body, and then some drug that might have been designed that plugs perfectly into a little cavity in this molecule. And in order to design a drug like this, you need to understand the shape and function of the molecule first. And I'm going to talk about how we tried to do this. What is shown here, this will come up a little bit later, is the number of structures, number of such molecules that have been solved over the years, and there is a big ramp up that coincides with the creation of some of the machines that are like an X-ray laser that I'll be talking about a little bit later. All right, so at first, if you think about this, you go biological molecules, people, life is very fragile. X-rays are in too large of a quantity are not good for you, right? When you go to the dentist, they make you wear a big lead suit to make sure that your internal organs are not exposed too much. So X-rays, not very good for fragile materials that make up life. Also, if you do a quick search online for X-ray laser, one of the first things you'll find is the type of original ideas for an X-ray laser that was more like a weapon, a space weapon in this case, where you could use an X-ray laser to shoot down other targets in space. So, we're talking about space weapons here. We're talking about X-rays that are fundamentally damaging to life. How does that mesh up, right? How does that make any sense to have such a machine to study life? So, how does something like this make sense? A space weapon and a big laser. How can you use that to see the molecules of life? So, hopefully, I can explain to you guys a little bit how that is possible. So, first, let's take a little step back, and a quick explanation of what X-rays are. X-rays are fundamentally like light, like the light that you see. So this is the so-called electromagnetic spectrum, really all different wavelengths of light. The visible light that we can see is in this range here, lengths of about a millionth of a centimeter or so. X-rays are just like light, but with shorter wavelengths. So this range over here is what we're talking about. X-rays are defined roughly as the range matching the typical spacing between atoms in solid materials. So that's why X-rays are a good tool to see where the atoms are. Their wavelengths matches the spacing between atoms very well. Most people are familiar with what X-rays typically do. They are very penetrating through soft material, and it allows you to see differences in density of material. This is the first X-ray picture ever taken back 124 years ago. So this is the hand of the wife of the man who first discovered those X-rays back then. So as I said, most people are pretty familiar with what X-rays do, and this is another typical human subject. This is actually me right here. So as you probably guessed, this is not me. But this is, however, me. So a little bit about me and my background with X-rays. So the introduction stated what I did as a PhD. So I was working with X-rays every day, trying to develop tools to help study biology, new tools. And this happened to be at the same time while I was in grad school. You can see a nice long fracture here, clean break, and 11 screws later, my arm is functional again. So no harm, no foul. And it was mentioned that I grew up in Quebec. I'm a French Canadian, so I'm a walking stereotype. I'm a French Canadian hockey player. So this is me at Illinois playing hockey, and this was after my arm was all healed up the following season. So, but yeah, that's how it happened. It happened to me while playing hockey. So, as I said, I'm a typical French Canadian hockey player. Big surprise. All right. So now light interacts with matter, right? You see me, you see other people here. Light bounces off surfaces, then we detect it with our eyes. Light will interact with matter in different manners. Some that we are very familiar with, like a reflection, light reflecting off of a mirror, for example. Light can also be deflected, passing through an object, like is shown here, and different wavelengths or different colors. Wavelengths and colors are the same thing. Will be deflected in different ways. This is called a refraction process. Light can also be absorbed and re-emitted, or re-radiated, means the same thing. Light can also be diffracted, bouncing off objects, and then we try to measure the angles at which they are deflected and understand the structure of the object that light bounced off. So those are typical interactions of light that allow us to develop multiple tools to study matter at the atomic scale. I'm going to talk mostly about diffraction and how we use that to deduce the shape or position of the components that are too small for us to see with our naked eye. Now, that was mostly a wave behavior of light, but light also behaves sometimes as particles. You cannot infinitely divide the energy of the light that you see into smaller and smaller chunks. A certain wavelength has a minimum size that is the smallest size, smallest amount of energy that it can be divided into, and we call those the particles of light photons. So when those minimal units of energy from light hit a material, and that amount of energy matches perfectly, say, the binding energy of an electron in an atom, then you can knock out this electron. So basically, the particles of light can come in and they can knock out some of the real particles inside the material, and then you can have energetic electrons that come out, and now those wreak havoc on the material, especially biological materials. That's what happens in your body if you get irradiated too much with X-rays. You knock out a lot of electrons from the atoms in your body, and then those walk around, bounce around, and they cause further damage to other parts of your body. So we call this in this case here, if you shine light on a metal and knock out some electrons, we call this the photoelectric effect. And one thing that surprises many people is this is what Albert Einstein won a Nobel Prize for, the photoelectric effect, not relativity. This is what he got a Nobel Prize for. So I'm talking about this here because we use the wave nature of light to study where the atoms are, but there's a consequence to that the same light that interacts with the sample also causes damage, and we have to worry about that when we do our measurement. We want to use the good properties of light to study the nature of the material without destroying what we're trying to measure. So this theme will come back again a little bit later. All right, so what we try to do, this is shown simply conceptually here, is we can picture every atom in a material as a slit, in this case, a hole in a plate where you have a wave of light coming in, and as the wave passes through a small hole or is diffracted off of an atom, it changes the distribution of the wave. In this case, it goes from a planar wave to a circular/spherical wave. So if you measure the pattern of light on a detector a certain distance away, you will be able to deduce that this is a spherical wave and that there was a single point object at this location. So it all starts from that very simple principle. If you have two such holes, or two atoms, now those two spherical waves, those two round waves here, are going to interfere with each other. It's going to change the pattern. If we can measure this pattern, we can again use that to deduce that, yeah, there were probably two different atoms that were this distance apart that are interfering with each other. So that's the principle we use to try to study where atoms are. Now it gets in detail a lot more complicated than this as you add more and more atoms, but the principle is the same. We have lots of these point sources that interfere, and we can deduce from that, from the way they interfere with each other, where the atoms are. So if you take this a step further and you have a real material, in this case table salt, that we're all familiar with. So sodium chloride is the chemical composition of table salt, where atoms of sodium and chloride are alternating in a regular pattern. We call this a crystal. And so if you have a crystal with a regular pattern like this, when the light gets diffracted off this crystal, it will create intense spots at very well-defined periodic positions. The periodic nature of the material will cause a diffraction pattern on a detector that is also periodic. And then we can use this pattern to deduce that the structure of table salt is a crystal with this alternating pattern. So that's sort of the first example of crystallography from a long time ago, from William Lawrence Bragg. And we call these peaks of intensity here Bragg peaks after him. And so that's what we try to do with biological material. We basically replace each of these points here that have a single atom with a very complicated molecule, like I showed a few slides ago. And now the pattern becomes much more complicated, but we can still deduce what each of these points are like and find where the atoms are within each of these points. So this is what we try to do in what we call protein or macromolecular crystallography, where we try to create a crystal from one of these molecules, a protein, a virus, or whatever. This can be a tedious process, trying to take a protein and crystallize it. It does not always work out, but if you can do it, then you end up with one of those diffraction patterns with lots of these spots here, these intense spots of more intense light, more intense X-rays. Then we can deduce computationally where the electrons are from this pattern. And from that, we can try to fit a model, an atomic model, where we know that this pattern looks like a certain pattern that we know exists in proteins or such molecules. And then we can build a model that best represents this structure. So that's what we try to do. And what is shown again on this plot here is the explosion of this technique of macromolecular crystallography. The blue part of the curve is what the structures that were deduced from crystallography, and the green and red were from other techniques. So crystallography has been a very, very successful method to try to understand where atoms are in the molecules of life. So it can be a little tedious at times, but it's very, very successful, and it shows by the hundreds of thousands of structures that have been deduced. What has caused this great explosion of structures in the late 80s, early 90s are these types of machines that we call synchrotron X-ray sources, synchrotron light sources. They are electron accelerators where electrons go around a ring, and as they curve around, the electrons get accelerated, and electrons that are accelerated will emit light. And if you build a machine with the proper parameters, the light that will be emitted will be at the X-ray range. So you can get all these beamlines, and you can dedicate a few to studying biology. And some of the bigger machines have up to 60 beamlines operating at a time, and they can be a kilometer in circumference. There's just a few examples shown here, and each of these, there's about 50 such machines around the world now, and they range from over a kilometer like these two to a little smaller. This one is in France, this one's in Chicago, for example. They are everywhere, and they're really the workhorse of understanding biology, understanding what the atoms are doing and where they are. And this is what such a measurement would look like. This is one angle. You have to rotate the crystal, this one crystal at the start of the measurement, and you have to rotate it to measure all different angles, but this is what one such pattern would look like. But as I said earlier, those molecules of life here are sensitive to X-rays. You cannot put them in an X-ray beam forever. They will eventually get damaged. And this is what we see at such a synchrotron source. As you expose the crystal longer, it starts to get destroyed. And this is what happens after a while, where it clearly has changed. You've clearly damaged the sample, and the high angle intensity spots disappear. You see these spots here go to a much higher angle from the center, and those start to disappear. And the higher angles give you the finer details. So as you start to lose the higher angles, you lose the information you're after. So as I said earlier, we use X-rays to probe the structure of matter of those molecules, but we want to do it in a way that does not destroy what we're trying to see. X-rays give us the information we want, but it also destroys the information we want. In order to minimize this damage, what is typically done is you freeze the crystal, liquid nitrogen. You freeze it to slow down the propagation of this damage. It still happens. It still will happen, but you can slow it down by making those damaged particles diffuse slower by freezing the motions out. This is not ideal for, this works great, as I said, but it's not ideal because you end up looking at the sample not in its natural state. Freezing something at liquid nitrogen temperature does not give you the natural environment of the molecule. It's not at the temperatures of life. It may freeze the sample in a position that's not natural, right? In the same way that, so, sort of like this, right? So you don't want to see Han Solo frozen. This is not in the natural position of Han Solo, right? You kind of want to see him chasing a stormtrooper or something. This is not necessarily telling you exactly what you want to get at, right? It gives you pretty good information. You can see his face and everything, but it doesn't tell you what it does, and it might be a little bit different at room temperature compared to freezing it. So, what X-ray lasers afford us in one way is to avoid the need to cool down the sample and look at the sample at more natural life conditions. All right, so this is what the X-ray laser looks like. Very different from the round machines that I showed earlier, right? So we have a synchrotron here on site. It is one of the small ones because it's one of the first ones ever. It's over here. It's relatively tiny compared to the very long straight accelerator that we use to make the X-ray laser. This accelerator has been here for 50 years or more. It started in the early 60s. Construction started in the early 60s. It has been repurposed about 10 years ago to use the last kilometer of the 3 kilometers to make X-rays, where we do experiments in 2 separate buildings where we use this X-ray beam. So, as I said, obviously a very different machine from one of those round synchrotrons that I showed earlier. I mentioned earlier that when you accelerate electrons, they will emit light. That is an unavoidable physical property of charged particles like electrons. So as you accelerate them or wiggle them, wiggling them like this is like accelerating them because you change the direction in which they're moving. Then electrons will emit some X-rays. If what makes an X-ray laser is different from other machines is how tight this bunch of electron is and how long these undulating magnets alternating now north-south poles that make the electrons wiggle back and forth. This is very long. This is 130 meters for us here. So as you start to emit X-rays at the beginning, those X-rays will re-interact with the electrons further down the line, and they will cause them to bunch up. At the end, it's very hard to see here. I apologize for that, but they end up with what we call micro bunches. And all these micro bunches, really what they are is, in the end, is that all the electrons move together rather than moving a little bit out of sync. They all move together and it behaves like one very large charge. And that one very large charge, when you oscillate it back and forth, makes a very, very intense short pulse of X-rays. So long story short here, we end up with a much brighter beam. These are the synchrotrons we talked about. These are X-ray lasers. 10 orders of magnitude more than before, which is 10 billion times more intense than previous, 10 million times more bright than previous sources. So all the X-rays come in a very short burst on the order of 1 millionth of a billionth of a second, and that allows us to do some unique measurements with such very, very, very short pulses of X-rays. As was mentioned earlier, this is the 10th year anniversary, and we had a party not so long ago. So 10 years ago, this was the first ever X-ray laser beam ever measured, and as I said, we had a big get-together where we reminisced about the good times and bad times, and I'm going to show you a few results of what we've been doing in the biology area using this machine. All right, so I still haven't quite told you why the most powerful X-ray beam in the world can work for studying fragile biology. And this is shown schematically here how this can work. Rather than have a very, very long pulse of X-rays, the pulses from those round machines that I mentioned are very long, like thousands of times longer than what you see here, this little pulse here. And so as you illuminate your sample with these X-rays, they start to change, and you start to measure a damaged sample later on. If you make this pulse very, very short, here, what is shown here are units of femtoseconds, and those are the millionths of a billionth of a second that I mentioned earlier. So very, very short. The X-rays can go through the sample before there's any time for anything to move. The simple conceptual way to picture this is the X-rays pass through the sample during this pulse at the speed of light, while the damage that happens, the changes that happen afterwards, happens at the speed of sound. The speed of light is 300,000 kilometers per second. The speed of sound is on the order of 300 meters per second. Very different. So the X-rays go through at the speed of light. The damage happens at the speed of sound. A little bit oversimplification, but for the most part, a good explanation of what happens. So very, very short pulses of X-rays, and then the sample is just obliterated. That allows us to put way more X-rays in this short pulse than we would be able to do in a long pulse, allowing us to get much better, clearer images by packing all these X-rays in a short amount of time that would not be possible to do if the measurement was longer. So for those of you who know who Kaiser Soze here, when I think of this, I think about what he says. He goes, and just like that, poof, it's gone. And it will never be seen again after that. So best movie ever, my favorite movie ever. So in practice, it looks like this. We have X-ray instruments where we deliver the beam to multiple systems to make these measurements. So those are some of the scientists setting up an experiment. And once in a while, even I do some work over there. But it looks very complicated to the naked eye, to the untrained eye, but it's conceptually very simple. There's lots of shiny metals, but it's actually very conceptually simple, which I will show on this slide here. So all we're really doing is we take the X-ray laser beam, so X-ray free electron laser beam. We take it, we use mirrors to focus it, to make it smaller, to take the X-rays and pack them into a smaller area. So we focus the beam to a certain point where we have a sample, in this case, being delivered in a flowing liquid jet. So we just cross the beams right here. And then we measure this diffraction, as I said, measure the angles at which the X-rays are deflected. They all come in on a straight line, and then we measure these, where at which angles those bright spots are, and then deduce the structure from there. So, as I said, conceptually very simple. We take the beam, focus it, hit the sample with it, measure it on a camera, an X-ray sensitive camera, not that different fundamentally from your iPhone camera. And do this multiple times to get all the different angles and try to get a structure out of that. What is shown here is what we call an LCP injector. It's basically a grease injector. It's that mimics a little bit the fatty membranes of cells. Some of the most important molecules in your body are at the boundary between the inside of your cells and the outside of your cells to let the right stuff pass through or react properly to external signals. So those we call those membrane proteins that are in the membrane of the cell. They don't like to live in water. They like to live in a fatty environment like the cell membrane. And this LCP here, this fat matrix, is a mimic of cell membranes. It creates an environment that's similar to that. And then the crystals can grow directly inside this matrix here. The crystals are not shown here, but they grow within these pores. And this fundamentally limits the size of the crystals. And so the smaller the crystals are, the more prone they are to get damaged quickly at one of those round machines that I talked about earlier, where you have a long illumination. You're putting a lot of X-rays on a small volume. It will get damaged faster. So this is perfect case use for our facility here for the X-ray laser. Crystals that are too small that we can deliver at room temperature in a basically native environment, much more suited to understand the true structure and true function of those molecules. So this is one of the results that I'm gonna show right now. So this is a recent result where we studied. This is one of those membrane proteins here. So this is a simple representation of one of those proteins. And this is a melatonin receptor. Melatonin is a molecule shown here. It's a small molecule that signals to your body that it's time to sleep or not, basically. So it is it is very common. You can buy melatonin supplements for dealing with sleep issues. Our modern world is not very conducive to having a good day-night sleep pattern with all that's going on and all the video games that you can play. And so, lots of people are suffering from sleep pattern issues, and these supplements here are one way to deal with it. But you can imagine designing a specific drug that is similar to that. This is the melatonin shown here in yellow inside of this protein that reacts to the signal from the melatonin that says it's time to go to sleep. And you can imagine designing a specific drug that will go do this job better and deal with certain sleep deficiencies better without causing side effects of having interactions with a different molecule that looks similar but not quite the same, and causing some bad reaction with some other molecule. So understanding the structure of these molecules and how they bind together allows people to design new and better drugs. And this is, billions of dollar industry for that kind of work. We were not the scientists that came to use the facility here for this particular work. They were not able to grow crystals that were large enough to be studied any other way than with the X-ray laser, and this allowed us to to understand the structure of two such receptors binding to melatonin. So there's receptor 1 and 2, and they are very similar to some extent. As you can see, the blue and orange ones are kind of similar, but they have some minor differences. And the work allowed us to understand how the melatonin goes in through this channel here in this case, and the second receptor, which has a slightly different function, has a second channel here that is less understood what it does. But this was studied with melatonin bound to it, as well as different types of drugs and other compounds, and it really allows us to understand how those two molecules interact with each other and potentially be the starting point for designing better treatments for sleep issues. So this would not have been possible without the very intense beam that we use here with the X-ray laser. Now, one of the other advantages of a machine like the X-ray laser is the pulses are so short, the measurements are so short, you can take a snapshot image of your object. Nothing moves during the measurement, so you can imagine starting a reaction by, for example, in photosynthesis, you can imagine starting a reaction by illuminating it with light, visible light, starting a reaction and measuring the changes with time by taking multiple snapshots. So this is what I'm going to talk about now, where I'm going to talk about photosynthesis. I'm going to talk about how how our atmosphere got its oxygen. So our planet a long time ago had a lot of water. This is two water molecules, H2O, so two hydrogens in white and one oxygen. If you have two such molecules and you put some energy in, you can produce two H2 molecules and one molecule that has two oxygen atoms in it. This is the air we breathe. The air we breathe has got about 20-21% oxygen in it, and our planet used to not have really any O2 in the atmosphere. And what has allowed our planet to become suitable for us to be here is this molecule here called photosystem 2. This molecule does a reaction like this, where it takes water and creates oxygen. And billions of, I don't know exactly, probably a few billion years ago, when this molecule showed up on our planet, it started to transform water and pump a bunch of oxygen in the atmosphere that allowed all the life that we all love right now to eventually flourish. And this happens by having 4 different photons, 4 different particles of light interact with the molecule sequentially to eventually take water and split it and produce oxygen. So, it can easily be argued that this is one of the most important molecules that on our planet for life here. It is believed to have originally started in bacteria, cyanobacteria, a long time ago, and eventually created the atmosphere that we have. And now it is everywhere in bacteria and all plants, and it continues to replenish our atmosphere with the air we breathe. So, very important molecule and a very important process that is not fully understood. Really, this four illumination, 4 light flashes cycle that eventually produces the oxygen we breathe is not well understood. And having the ability to look at these molecules at room temperature, not frozen, with flash photography of the X-ray laser allows us to try to see what happens in this reaction here. And so I have a little movie here that will sort of show how we do this. And so this is the photosystem 2 molecule. Inside there's a manganese calcium cluster where all the action happens, where there's a few atoms of manganese and calcium. And we can either deliver the crystals inside of a liquid jet or shoot them on a little tape drive here and then illuminate them with green light, either 1, 2, 3, or 4 flashes to go through this cycle, and then eventually shoot them at a certain delay after this light illumination to see what state it is at that certain point. And that allows us to, right now, to have the beginnings of a molecular movie of how this molecule takes water and splits it to produce the oxygen we breathe. So this is a very important prototypical example of what we can do in terms of measuring room temperature dynamics of important molecules to life. All right, so those are the only 2 examples of the biology that we do that I will show. And then I will show you what I consider spectacular movies of what happens after this X-ray illumination. As I said, the X-rays come in, they completely obliterate the sample, but amazingly, you still have time to take the measurement that you want, even though this happens after your measurement. So this is a liquid jet that you'll see that flows from right to left. This little white line here is actually a flash of light because the jet gets so hot. That's picked up by the camera. And the jet flows this way, and the X-rays are going to hit here, and you'll see what happens like a millionth of a second after the X-rays come, and yet we have a clear image of the sample we're interested in. So the X-rays come in, and they basically cut a hole in the jet that's 100 times larger than the X-ray beam itself. So it completely vaporizes it and explodes it. And yet, as I said, all this happens after you've been able to make a successful measurement of a very fragile molecule. So this is the beginning of the process here, where the X-rays come in, they vaporize the water, gets very hot, and then this creates a very high pressures that push the water away, and then eventually it forms this big gap in the jet. So as I said, amazingly this happens even though we're able to make a real measurement of the very fine details of the position of the atoms we're interested in. This gap in the jet here is millions of times bigger than the features we're trying to measure. So, and this is part of the original figure that I showed. I really need to acknowledge Claudio Stan, who has now moved on to Rutgers University. This all happened, these types of measurements happened all because of him. These beautiful movies were really all due to him. And so the X-rays come in, they vaporize some water, and the water causes the jet to separate and explode. Now, if you do this in droplets, you get a similar effect. But I remember before we made this experiment, I predicted that the droplet that gets hit by the X-rays, this one here, would be completely obliterated, but the next neighbors would be perfectly fine. And you will see how wrong I was about that. So the X-ray beam completely blows up that droplet. It's completely gone. And the pressure wave pushes the next neighbors to the point that if you keep play the movie for a little bit longer, the three closest droplets eventually all get absorbed into one droplet. So this is the beginning phase here where the X-rays hit, vaporize the water again, and then a big shock wave propagates and eventually completely pancakes the neighbors. That was unexpected, at least from me. And actually not just from me, because previously to that, a couple of years before that, we had a measurement on similar water droplets where we were interested to look at the water structure itself, completely unrelated measurement. And this is what people thought happens to a droplet when you hit it with an X-ray laser beam like this, where the water is just shooting out the side that you hit. And this even made the cover of a very, very reputable journal where we just got this artist rendering wrong here. A few years later, we showed that that's not at all how water droplets explode when you eliminate them with an X-ray laser beam. All right. One more movie for pure entertainment. Our very talented communications department here made a movie of a collection of all these exploding jets, pretty music. I'll just let you enjoy it for 40 seconds here. So, not to dwell on the point too much here, but what I find amazing is all this happens, and yet we see where atoms are, as I said, a million times smaller features than these giant explosions. All of this can be done despite this incredible changes that we see in the sample long after the X-rays are gone. As I said, the X-rays pass through the sample at the speed of light. This happens more at the speed of sound. Pretty amazing to me still that this works. Now I'll take the time scale even shorter here. Those movies happen on the millionth of a second time frame. We're going to go the millionth of a billionth of a second time scale here and try to see how the sample can change during the time scales of the X-ray exposure. This is a simple cartoon of how a sample can change under X-ray illumination. You can picture two different kinds of damage or changes with time. What we call global damage, where everything just flies apart in sort of a uniform manner, random uniform manner. And then after a certain time, when your crystal is undergoing such a process, you don't have a regular arrangement of molecules anymore. You don't have diffraction anymore because you don't have a regular array of objects to diffract from. So in that case, your sample will diffract for a while. When it's too damaged, it just won't diffract anymore. You could also picture different behavior where not all the atoms are the same, right? Some atoms are are heavier than others, and they will absorb X-rays more than others. And you can picture that these areas will be more specifically damaged than others, and they will change faster than others. And you'll still measure a diffraction pattern that can be interpreted, but some parts will change, some parts won't. And now you have to figure out at what point our assumption that the sample is not changed during the measurement is no longer valid, right? So we devised an experiment to try to figure that out. As I said before, X-rays that we use to understand the structure of matter also cause changes in it. And 9 out of 10 X-rays that we shoot at the sample actually cause damage. Only 1 is diffracted for every 10 that gets absorbed and causes every 9 that gets absorbed and causes damage. So it is a big concern as we make the X-ray beam smaller and smaller to look at smaller and smaller particles. So as I said, we devised an experiment to try to study what happens on this femtosecond time scale, or the millionth of a billionth of a second time scale, where this looks pretty similar to what I showed before. We focused the X-rays, in this case, to a spot that's 100 times smaller than what I showed before. But we can use the X-ray laser to make 2 pulses that have slightly different colors or different wavelengths of X-rays. So a blue pulse and a red pulse, and we can control the time between them from 20 femtoseconds to 100 femtoseconds. Again, femtoseconds being a millionth of a billionth of a second. And we can separate them in color enough that if we use, in this case, an iron filter after the sample, we can have the blue X-rays that come first be here and be totally absorbed by the filter, and the red X-rays go through. This is the transmission through this iron filter. Here you have about 30% transmission, and here you have essentially 0 transmission. So that allows us to damage the sample with the blue X-rays and measure the structure of the damaged sample with the red X-rays. So compared to the movies I showed before, the time scale of this is a billion times faster, basically. So that is the type of measurements we need to do to understand if our assumptions that the sample is not damaged are valid or not. So we did that on a molecule that contains multiple disulfide bonds, so 2 sulfur atoms that are next to each other. Sulfur atoms are present in most molecules of life, and they are heavier than the more typical carbon or oxygen atoms that are more prevalent, and so they are likely to change more during the X-ray measurement than other parts of the sample. And so this is what is shown here, where we see a 6-frame movie of what happens. This is what we see when we have no delay between the 2 colors, and this is what we see as we increase the time delay all the way to 100 millionth of a billionth of a second. And basically what is shown here is in green is where the electrons are, or where the sulfurs are, and in red is where they used to be, by making a difference between the 0 delay and the longer delay measurement. So as you can see, they just fly farther apart. As I said, green is where they're going and red is where they used to be. So with time, those 2 sulfur atoms are clearly flying apart, while the rest of the molecule is not changing that much. So that definitely challenges our assumption under very, very intense, very tight, small X-ray illumination, whether there's no change in the sample or not. So if you extract the distance between those 2 sulfur atoms as a function of time here, you can see that they fly apart somewhat linearly in a somewhat straight fashion. Our typical X-ray measurements are about 30 femtoseconds here, and you can see there's definitely a change, a 25% change between in the distance of these 2 atoms, and that is very significant if you're trying to understand the detailed mechanisms of what this molecule does. So this is a big error if you have not accounted for it. And if you extrapolate this line and get a speed by taking this distance dividing by the time, you get roughly the speed of light. So sorry, the speed of sound. As I said earlier, the damage kind of happens at the speed of sound. So I like this pattern here because it reminded me of Mickey Mouse the first time we saw it. Anyway, so as we try to make the samples smaller and smaller, getting rid of the need to make crystals and trying to do this with single molecules, we need to pack more and more X-rays into a smaller volume, and these types of structural changes during the measurement become more and more of a concern. This is another movie of the ultimate experiment that we would like to do, that we have attempted also a little bit already. The X-rays come down the X-ray laser machine. Here we have single molecules, single proteins floating in air, being sucked into vacuum, where we try to interact them one at a time with an X-ray beam, measure a diffraction pattern, similar to the crystals I talked about, but now single molecules. So the X-ray focus has to be a lot smaller. We do this a bunch of times, tens of thousands to millions of times, depending on who you ask. And then you figure out the relative orientation of all these patterns and try to build your 3D, three-dimensional scattering pattern from this data. And then you press the magic button on your computer and you get the structure at the end. So this is basically what we do with crystals now, but we would like to do this with single molecules. First, work our way towards smaller crystals, then eventually single molecules. And that requires, as I said, a much tighter X-ray focus that will be more prone to causing damage. So we really need to understand those dynamics of change during the X-ray illumination if we are ever to understand the results of such an experiment at the atomic level, seeing where every atom is and trusting the results. So, as I said, we've done some work in this area. This is very reminiscent of the movie that I just showed. You see these individual slices of the diffraction pattern. You piece them together and eventually try to get a structure. This was a single large virus. As you can see, the pattern is not complete. There's lots of gaps between these each of these slices. And so what I'm going to show you next is what we're doing to try to resolve the data sparsity problem, as well as I already mentioned, we want to understand the damage mechanisms. So we are about 80% through the construction of a new accelerator and a new system that we call LCLS2. So this is a reverse view of the picture I showed earlier of LCLS. We currently use the last kilometer of the accelerator. The first kilometer has been refurbished to be a superconducting accelerator. It will come online in 2021 and it will produce 1 million pulses per second. Currently we operate at 120 pulses per second. So a huge increase in number of pulses. And now we might be able to do these very challenging experiments that require millions of images to obtain structures from smaller and smaller objects, eventually potentially single molecules. So this is sort of the next step, and we are in the middle of a 15-month shutdown now, where we're not running any experiments to complete this construction here. So a very big, important project for us. All right, before I summarize this, just wanted to quickly say that I don't do this all by myself, obviously, and those are some of the important institutions that contributed to the results that I showed here. So, all right, so as a quick summary here, I hope I have shown you that X-ray lasers allow studying molecules of life in action with improved resolution compared to more conventional X-ray sources. It has medical applications and understanding photosynthesis, among many others. I only showed a couple, but those two, the photosynthetic cycle, as well as understanding medically relevant molecules and how they bind to drugs, are two very important areas that we are pursuing. The sample is completely destroyed, but only after the image is captured. And that is really amazing, in my opinion. As we try to move towards single molecules and making molecular movies, we require smaller beams, which can lead to more sample damage, and more images, which we can start to address with the higher repetition rate of LCLS2 with millions of pulses, 1 million pulses per second. And more studies are needed to understand where our assumption that there is no damage fails. I hope some of the results I showed drove that point home for you guys. So that's about it. So this is what we do here. This is what me and a few of my colleagues do. We basically spend our days trying to improve device methods to understand the molecules of life in action. And this is what we do. And this picture here shows a good summary of what we're trying to do. We're trying to get a structure of a molecule by flowing little crystals inside a jet that gets completely destroyed by our beam, but not fast enough to prevent us from performing these measurements. So as I said, this is what we do. We study the molecules of life in action. Every day we come to work and we drive towards that goal. So, and thank you.
Okay, we have some time for questions. Please raise your hand, get recognized. You each have a microphone in front of you. So after you're recognized, you push the button on the microphone, it will light up red, and then you can be heard, and also you'll be on the videotape. So questions, please.
So the fact that the damage propagates at about the speed of sound, is that just an empirical experimental finding, or is there some theoretical basis for it?
Right. Well, the sound is basically the motion of atoms, pushing on each other, right? So damage would be the same thing when atoms fly apart. It's atoms pushing on each other, and there's a certain limit to how fast they can push each other. So that part is roughly right. What is not right is the subtle electronic changes to an atom. Each atom has got a cloud of electrons around it, and those electrons fly apart much faster. So a certain atom can lose an electron much faster than the speed of sound, and that would be a much more subtle effect than the physical blowing up of the object. So those two, the electronic damage is more what we're concerned about than them flying apart at the speed of sound, which, as I said, is mostly right because that's how fast things can push each other out of the way.
Do you have to worry much about detector damage from the X-rays?
Yes, but maybe not for the reasons you might guess. The signals we're measuring are fine. The detectors are pretty well matched to the intensity of those spots of diffraction that we measure. What we have to worry about is completely different. Sometimes we shoot these jets into vacuum, and a water jet in vacuum, if you do something wrong with it, they will freeze. The water will turn into ice. And the diffraction from ice crystals is much more intense, like a million times more intense. And that completely destroys our detectors sometimes. When the experiment is running properly, no, we do not have to worry about damaging the detector. We do put a small shield in front of it to capture the debris that flies at it. So that physical damage could happen if we didn't have a debris shield. Yeah.
I guess I've got to add, the accelerators, of course, come from high energy physics, and also the detectors come from high energy physics. So those materials have seen much worse than what you get in these experiments. Other questions? Please.
When you show that sequence of images representing the explosion of the sample, I noticed that most of all have a pattern of a double cone, let's say. But there are a few that are different. They seem like a fuse, something like that. I remember a green image, for example. It was like
right.
that.
Yeah. Okay. So that comes from. Let me see if I can. Let me grab an image. Let me pause it in a spot that would show it. All right. Let's see. Okay, yeah, this is a good one. Okay, yeah. So what happens here in this case is you can only make a jet as small as the tube that the water flows in, all right? That makes a wall and the jet comes out at that size. And that's limited in size by the smallest tube you can make that will not clog with whatever piece of dust. So that's usually about 20 microns or 20 millionths of a meter. All right. We can get around that by making a virtual tube, by flowing gas around the liquid, and then the gas makes a virtual tube. And if a piece of dust comes out, it just pushes the gas out of the way, and then the gas tube reforms. And this is what's happening here. There's a co-flowing gas that basically folds this cone back, because it's basically wind is folding that cone back. Right, right. So it starts out with a cone on both sides, but then the wind that makes this jet a lot smaller folds the cone of debris back away from the nozzle.
Way in the back.
What other experiments do you do besides these atomic structure measurements?
You asked what else? What else do we do? Is that
Yes. What all other experiments do you do that could be of interest besides just molecular experiments?
well, LCLS does work in all sorts of areas: material science, plasma physics, just chemistry as well, ultra-fast chemistry, some fundamental atomic interactions, trying to study the energy levels in complicated atomic systems, binding energies and things like that. So we have biology is one of 7 roughly major areas of work that LCLS, the X-ray laser, pursues. I hope that answers your question.
I think if you go to our public lecture website, you'll find other lectures on some of these other applications, magnetism, movies of chemical reactions, etc. So have a look for that. Greg. Use your microphone.
From what you explained about the damage, how will it work for multi-bunches in LCLS2? Because the first bunch will destroy the sample. The second sample won't have anything to look at.
Right. So that is a very good question. Those movies that you see here, they go on for longer than the time for the next pulse if we go a millionth per second. That's certainly true for the big jet that I showed at the beginning. These smaller jets here, you can accelerate them fast enough to be able to push everything out of the way before the next pulse comes at a million pulses per second. You have to run them a factor of 5 faster than this particular jet that you see here, for example.
So you're saying the samples will move so fast that the next sample will be there for the next pulse.
Right. That is a hard requirement for these types of measurements.
And
you have to push the sample that's damaged out of the way to get to an undamaged part of a jet before the next pulse comes. And some jets or droplets are not suitable for 1 million pulses per second. They are too slow for doing that.
So let's just take a couple more questions. There was one over here.
Yes, I had a question about the membrane protein experiment with the melatonin receptor. What is the resolution you're getting in those, like 1 or 2 angstroms?
Right.
Or something?
Don't quote me on that because I don't remember the exact number. It's on the order of 2.5 to 3 angstrom. So an angstrom is 1/10 of a billionth of a meter. So typical atomic spacing of say a silicon in your computer chip is about 4 angstrom. So this is resolution smaller than your typical silicon to silicon atomic spacing.
And my other question was, and this may not be applicable to what you do, but have you thought about looking at a mixture of proteins by coupling your injector to like a liquid chromatograph where you can separate a lot of samples and then have like another dimension of separation in time and introduce them to your beam at different times and then have many structures solved in a single run?
Yeah,
perhaps?
absolutely. I didn't show this, but this is an area of active research to look at dynamics from rapidly mixing 2 things together rather than using light to start a reaction. Not that many things in life are light sensitive. You and I are not particularly light sensitive, most of our body. Most of the reactions happen by two things reacting with each other. And rapid mixing is, we need to develop better mixing sample delivery devices to really exploit that fully, but there's been a few papers about that already, and that's an area that we expect will grow more.
Thank you.
Last question over here.
Thanks for speaking. I like the jokes. Is there any direction or interest in applying this methodology to neuroscience, and how far away would that be?
Yeah, there's been one paper published a few years back looking at certain molecule involved in synaptic reactions. So it's really this is applicable to any structural biology problem as long as it's a problem that really requires the unique capabilities of an X-ray laser. A lot of these problems can still be done at those round synchrotron sources, but if the crystals are too small, or if there can be some dynamic mixing involved, then we can absolutely be of help for that kind of a problem. Yeah.
Where did the paper come out of?
It came out of Axel Brunger's group here at Stanford, so you can Google him. Yeah, he's a professor here at Stanford.
Well, very good. Thank you very, very much. After the lecture, Sebastian will be out in the lobby. You can ask him more questions informally. Until then, let's thank him very much. And we hope to see you all at the end of July for the next round of these public lectures. So thank you.